Hello, welcome to this course that will introduce you to the emerging field of systems biology. This course can be taken as a standalone course that will give you a brief overview of this new discipline in biological sciences, or it can be taken as a part of a 5-course series that over a year can lead to a certificate in systems biology. So what is systems biology? Over the past 50 years or so, we have gained a really good understanding of the various parts and components of what may of our cells, tissues, and organs, starting from genetics and genomics, where the genes of many organisms, including that of humans, have been fully decoded and sequenced, to biochemistry, where the proteins of where many proteins have been studied for both structure and function, and in depth to understand what the proteins do by themselves and what they do by interacting with their partners, to cell and molecular biology, where we study how proteins are organized to form subcellular systems such as the mitochondria or nucleus or the cell cytoskeleton, to physiology, which is focused on the study of functions of cells, tissues, organs, and organisms. There has been a vast amount of knowledge that has been gained. This knowledge sort of allows us now to gain a perspective where one can start at the level of genes and go to the level of functions at the cell, tissue, and organismal level, and understand how the information in genes are decoded to form proteins, and how proteins interact with other components of the cell, such as lipids and sugars and nucleotides and so on, and all of these together give rise to cellular, tissue, and organismal functions. It is these kind of integrated study that is called systems biology. So the word systems itself is an amorphous term. It can mean systems at various levels of biological organization. So one might have a system at the level of a cell, or at the level of a tissue and organ, or at the level of the whole organisms, or going back to the other end, it may be subcellular levels, like I said before, either mitochondria or nucleus can also be called systems. So there isn't one fixed definition of what systems biology will be or is, and different people can have a different perspective of how the field is growing and focused on. In this course, I will focus largely on systems biology at the cellular level and mostly mammalian cells, because it provides us a natural segue into how cells become tissues and organs, and allow me towards the end of this course to describe briefly systems approaches to the study in medicine and pharmacology and therapeutics. I've organized the course, so there are many ways to organize this course, and there probably is not any one correct or wrong way to do this. So I've organized this course to sort of follow the natural flow in which the various biological disciplines sort of grew and came to be. So the course starts with sort of molecular components, biochemistry and cell biology, how individual components, genes, proteins, etc. were discovered and their functions analyzed, and how these were put together to form smaller subcellular systems, such as a signaling pathway or a metabolic pathway or cellular machines such as the cytoskeleton or the electrical machinery, the groups of channels in the cell membrane, and how these were studied both experimentally and how they can be studied computationally using dynamical models to sort of both understand function and predict future behavior. I then move on to describing newer technologies, the omics technologies that allow us to study changes in many components at a time. These kinds of omic technologies, genomics and proteomics and so on, have become the hallmark or sort of the defining characteristics of the experimental approaches used in systems biology. And these technologies allow us to do survey type experiments where we can study how many components, so for in the case of gene expression or microarray experiments, how levels of many messenger RNAs change in response to one or more perturbations. Similarly, proteomics allows us to study how many proteins may change in response to a certain kind of perturbation. These large numbers of changes require, and the large data sets that come from these omics experiments require us to organize these data in an appropriate manner that can be used for analysis and extract knowledge. And sometimes these large data sets are now increasingly being called big data, and the field that is devoted to the analysis and organization of such data is called bioinformatics. And these data can be analyzed using a variety of statistical and mathematical tools, and the field of mathematics that deals with this kind of analysis of large data sets is called graph theory or network analysis. And we will study we will learn how network analysis has been very useful in understanding how systems are organized. Finally, I will end with some brief description of how systems biology or systems approaches are going to be very useful in the field of medicine and therapeutics. As this is an online course, there are not going to be any experiments to do, and also I will not really have you run any simulations or do any network or computational analysis. Largely, this is going to be a thinking course rather than a doing course. By this, what I mean is that I want you to get a sense of how one can use quantitative reasoning to deal with large data sets, and how understanding what kinds of mathematical representation are appropriate for different kinds of biological questions and subcellular systems, and how mathematical analysis can provide both deep understanding of how behaviors occur and emerge, and how one can get predictive value from computational analysis. This course, of course, will be followed by three other courses, one focused on experimental technologies and systems biology, which will really be, if you want to call it, sort of a show and tell course, where we will describe to you how these various technologies are used and what they can be used for in the context of systems biology experiments, and a course on bioinformatics, graph theory and network analysis, and a course on dynamical modeling. I hope you will find all of these courses both interesting and useful, both in understanding contemporary biology and in sort of bringing in cutting-edge technologies to your own research and career goals. So let us go over the key objectives of this course. As you take the course, you might find that some parts of the course are simpler or more difficult depending on what the material is and what your background is. And you might be wondering, why is this sort of material placed at this particular juncture? As I said before, there's no really right or wrong way of organizing this. One can do it in several different ways, but irrespective of how the material is sequenced, overall, when one puts together the material that I will present to you in this course, it should satisfy the following course objectives. One, learn about low throughput and high throughput ways by which systems components have been identified and studied. Two, learn about small scale dynamical models using differential equations and what these models can tell us about systems behavior. Three, learn about network analysis and how it can be used to obtain an understanding of systems level organization. Four, learn how interaction between components lead to the emergence of systems level properties. And five, learn about the potential for systems level reasoning in medicine and pharmacology. Together, I hope this will sort of take you to a understanding of biology or cell biology, moving from components and interactions into sort of groups or sets of components that are associated with a function, and how these subcellular functions give rise to cell and tissue level functions.